Hello, welcome to 13 Universal. This is A-level Caie biology. We continue with cell membrane topical series 3 as we prepare towards the paper 1 left in 11th of June 2024. So this is Shedro, your teacher. Okay, so come with me. Let's dive into cell membrane questions to demystify this topic. All right, look at the first one there. Just want to wish you the best in your exams. Okay, but remember, the first thing you just understand the question is that the cell surface membrane structure is described as fluid mosaic. Okay, let's go. Which statement describes the mosaic part of the cell surface membrane? Okay, so which of them? So I would like you to pause the video and try to solve it. Okay, remember, the fluid talks about the movement of phospholipid molecules and of course protein, but especially movement of phospholipid within their monolayer. That's fluid. Okay, mosaic means that the protein molecules are arranged in a scattered or interspersed pattern. Okay, let's go. A says the different. We're looking at what explains mosaic. The different patterns that are obtained by the moving phospholipid molecules. Now, this moving is for fluid, so it's not for mosaic. All right. B says the random distribution of cholesterol molecules within the phospholipid bilayer. Now that mosaic talks of protein, not cholesterol. Is that okay? C says the regular pattern produced by the phospholipid heads, and now that pattern of distribution is only for protein when we deal with mosaic and not, all right, for phospholipid. And that brings us to option D, the scattering of different proteins. Yeah, that's it, within the phospholipid bilayer. The scattering of different proteins explains mosaic. Okay, while the movement of phospholipid explains fluid. All right, so the answer of course is D. Let's go. Now it says solute X. Now this is called solute X is at a higher concentration outside a cell, all right, than in the cytoplasm. So we have more of it, all right, on the outside of a cell than the cytoplasm. So which processes may allow solute X to be moved through cell surface membrane? Okay, which process may allow it to be moved? Okay, so these are the processes. I want you to pause the video, okay, and then try to solve this question. Pause the video. Now I'm going to show you, all right, a shortcut. I'll show you the long way to solve this question. I'll show you a shortcut to solve this question. If this were to be a cell, let's assume, okay, and they said that the solute, all right, was of high concentration. Look at it here, higher concentration outside of a cell than in the cytoplasm. Let's assume that, right, this is the solute here. Okay, it looks like this. That's solute X. And then we have it outside here. This is outside the cell. Okay, so we have more of this solute outside. That's why the concentration outside is more. So how can this solute move? Remember that, okay, it could move depending on the nature, whether it is polar or not. But if it's moving from outside to inside, it's moving from high concentration to low concentration, so which is diffusion. Okay, if it is not charged, right, if it's non-polar. But if it is polar, we'll call it facilitated diffusion, right? So if it's moving from inside to outside, that is moving from low concentration to, all right, to high concentration, that's active transport. Did you get? Yeah, active transport. Okay, all right. It's possible that if it is, of course, it's a solute, it's not bulky. So if it is bulky, all right, we could mention exocytosis or endocytosis, but this is not bulk. We're dealing with solute, so that's the way. So options that have active transport, diffusion, right, and of course that have diffusion, facilitated diffusion will be the answer. So if you check it, you see that that option is A. Okay, now it can never be, all right, B, C, D, because we have B has osmosis, C has osmosis, D has osmosis. We are dealing with a solute, and osmosis deals with the movement of water molecules. Okay, not solute. Okay, this solute will never move by osmosis because it is not water. Osmosis is specifically for water molecule, and that's why the answer is A. Is that okay? All right, 3 molecules found in prokaryotes are listed. Here are they: phospholipid, protein, peptidoglycan. The question says, which molecules are found in prokaryotic cell surface membrane? Cell surface membrane. Which of them are found? So I just want you to pause this video and try to solve the question, guys. Remember, your exam is around the corner. We practice questions here. So which molecules are found in prokaryotic cell membrane? Okay, yeah, phospholipid is part of cell membrane. Okay, protein is part of cell membrane. Peptidoglycan, is it part of cell membrane or prokaryotic cells? Of course, no. Peptidoglycan is part of cell wall, not cell membrane. So option should have 1 and 2. So which option has 1 and 2 only? Of course, it's B. Okay, so let's go to the next question quickly. What happens to the surface area to volume ratio? This is ratio, not ration, of a cube when the length of each side is doubled? So what happens when the length of each side is doubled? When it is doubled, so I want you to pause the video, try it first. Don't check the mask and try it first. Remember that surface area, okay? This is the principle. Surface area to volume ratio, right? This surface area to volume ratio, okay? Yeah, to volume ratio. By principle, surface area to volume ratio, this surface area over volume, okay? By principle is indirectly proportional to length. Okay, that means the longer all right, the cell diffusion pathway, right, of course, of the organism or the cube, okay, the lower the surface area to volume ratio. Okay, so but the shorter the length, right, the bigger this. So if the surface area to volume ratio of a cube, if the length of each side is doubled, so that means that there's the opposite, okay, inversely proportional. So if the length is doubled, surface area is going to be halved. Okay, so it's going to be halved. Okay, all right, so the option that has half there, right, is going to be halved. Okay, and then that is B, because they are inversely proportional. Is that okay? All right, so let's go into other questions. Long question says full size potato were placed into test tubes containing equal volumes of different concentrations. So they vary the concentration, different concentrations of sucrose solution, and left for 30 minutes. All the variables were controlled. After 30 minutes, the potato piece in one of the concentrations of sucrose solution had not changed in size. So if it has not changed in size, what is that? Okay, it has not changed in size. So what can be concluded from the result for this potato piece, guys? All right, so these are the statements. I want you to pause the video and try to solve the question. I believe in you. I know you can do it. Okay, so after 30 minutes, the potato piece in one of the concentration of sucrose solution had not changed in size. This means that this potato must have been put in a solution of sucrose with same water potential, right? So look at it. So let's look at the statement that could be concluded. One says the concentration of sucrose in the potato is the same as the concentration of the sucrose solution. No, that's not correct. Why? All right, because if it remains the same size, that means that is the water potential we're looking at, not sucrose concentration. Is that okay? So the water potential should be the same. Okay, look at number 2. See, the water potential of the potato is the same as that of the sucrose solution. So this is correct. If it must remain, if it had not changed in size. Okay, so this is correct. That means the water potential of the potato is the same as water potential of the sugar solution. Then the third statement says there's no movement of water into or out of the potato. That is wrong. All right, there is movement, but there's no net movement. Of course, if you put it inside this, right, this potato here, all right, that's called isotonic solution. That's what it's talking about. So the movement of water, right, inside the potato and the movement outside the potato, all right, the movement is the same. So there's no net movement, but there's movement of water even in isotonic solution. So that is why the size does not change. Okay, so it is only 2. That's correct, and that's D. Okay, 4 agar blocks. Come with me. Let's run. 4 agar blocks, can see that, are stained green using universal indicator and suspended in 1 mole per dm cube hydrochloric acid. So, all right, you can see it's suspended here. Okay, that's the agar beaker hydrochloric acid with universal indicator. The time taken for the agar blocks to change from green to red was recorded. Okay, which agar block would be the first to become completely red? Okay, so which one will give us the highest rate of diffusion? Okay, all right, so these are the options. So I want you to pause the video and solve the question. Okay, so somebody figures out that the one that can give us the highest rate of diffusion, okay, is the one that has higher surface area to volume ratio. Now if you try to start calculating surface area and volume ratio of any of these, it's going to take you time. Here is a trick on how to solve this question. Here's a trick here. First of all, look at the first dimension. Remember that, all right, whether volume, of course, if you're going to calculate volume, of course, surface area is usually length times width. Okay, length times width. All right, so though you put n here, n is the number of surfaces, right? All right, all over. Now length, volume is length times, okay, width times height. But if you try to do that, because the one that will give us the fastest is the one that has the largest, the highest surface area to volume ratio. Okay, surface area, the one that gives us the highest surface area over volume, that's this ratio, okay, will give us the fastest, right? But if you try to do calculation, it's going to take you time. So how do you do that? So come to the dimensions. Remember that dimension is length, width, height. So check it. Length, now they have the same length, so the one that has smaller length will give us the faster. Remember, the shorter the length, okay, the higher the surface area. Now but you see, option A, option B, look at it. Okay, so let me show you how to do it, so that next time you don't waste time with this question. This is length, this is width, okay, that is height. All right, the same thing here, length, okay, width, all right, height. The same thing here, length, width, height. Don't waste your time solving this question. Just next time you see it, I'm going to give you a secret now. So but just know the dimension. These are the dimensions. Okay, so what you do first, guys, is start from the length. Okay, now all of them are the dimensions. So let's check the ones that have the smallest length. About 3 of them have the smallest length, which is 2 cm. Okay, leave it. We'll go to the width. Which one has the smallest width? A has 2, B has 1, C has 3. Okay, this has 2. So once you get to the one that has the smallest width, that's the answer. But if all of them have the same width, go to the height. The one that has the smallest height, that's the answer. So the one that has the smallest dimension, either length or width, the first of all, we'll start from the length. The one that has the smallest length is the answer, or the one that has the smallest, if they have the same length, the one that has the smallest width is the answer. If they have the same width, the one that has the smallest height. So this has smallest width. Okay, so that's B. So don't waste your time. The answer is B. Okay, all right. There are a lot of questions like that. Okay, look at number 7. An indicator mixed with agar forms a pink color, and The pink colored agar becomes colorless when put in acid. When put in acid, it becomes colorless. Blocks of pink colored agar are cut to different sizes and put in acid. Okay, variables are kept constant. Which block becomes colorless most quickly? Look at it, guys. These questions comes out every year. So let's solve the question. Now look at it. Pause the video and apply the concept I taught in the last question. Pause the video, okay, and then come back. Now look at this. So look at this is the length. So which one has the smallest length here? A. It doesn't matter other dimension. Just start with the length. The one that has smallest length, all right, is A. 3 millimeters. This is 6 millimeters. B is 6 millimeters. C is 6 millimeters. D is 12 millimeters. So assuming that all of them have the same length, you go to width. The one that has smallest width. If all of them have the same width, you go to height. All right. But here, the first to have the smallest length is A. So A automatically will give us the higher surface area to volume ratio. The shorter the length, the higher the surface area to volume ratio, and the faster the rate of diffusion. Okay, because the one that becomes colorless most quickly is A, because it has the smallest length. Is that okay? Don't waste time there. Don't start doing the calculation, because the identical plant cells were put into one of 3 different concentrations of sugar solution. So you can see that. Okay, one is 10, that's 10%, 5%, 2.5%. Okay, so let's go on. The cells were left for 15 minutes and then observed using a light microscope. Yeah, let's see what we have. Cell X, cell Y, cell Z. See them cute cells. Okay, which statement is correct, guys? Yeah, which statement is correct? The one that is correct. Oh, which statement is not correct? I almost missed it. Okay, yeah. So the question is asking which statement is not correct. That means that 3 of them are correct. 1 isn't correct. 1 is not. So which one is not correct? Okay, let's find it. But I want you to pause the video and try it by yourself. Which one is not correct? So we have cell X. See, cell X has the same water potential as the sugar solution it was put into. Is this correct? Yes. Can you see it wasn't turgid, it wasn't plasmolized, just the same. So this is correct, right? Yeah. Okay. Cell Y is turgid. Yeah, you can see it's turgid here. You can see the vacuole swells up. Okay. The cells also swells up. So cell Y is turgid, and cell Z is plasmolized. Here you can see cell Z plasmolized. How do we know? The cell membrane is withdrawing from the cell wall. You can see this is cell membrane withdrawing from cell wall. Okay, so that's plasmolized. So statement option B is correct. We are looking for what is not correct. Okay, C says cell Y was put into the 2.5% sugar solution. So the one 2.5 is the one that has the lowest concentration. Okay, so that means it will have the highest water potential. So if it has the highest water potential, so more than cell Y, water would have moved from this solution with low solute concentration but high water potential. So water moves from this solution into this. That is why it is turgid. So A, B, C are correct. So D must be the answer. Now let's look at D. Cell Z had a more negative water potential than the sugar solution it was put into. Cell Z had a more negative. What does it mean to have a more negative? A more negative means to have low water potential. More negative means low water potential. So anytime you see more negative, okay, it means low water potential. That cell Z had low water potential, low. Okay. Now, if it has low water potential than the solution, that means water would have moved into cell D. Okay, and it would have become turgid instead of being plasmolized. Okay, that means that this statement is wrong. All right, it's a wrong statement. That's the one that is not correct. So cell D, cell Z, okay, definitely had high water potential. That's why it lost water and became plasmolyzed. Okay. So the answer is D, guys. Let's roll. Which roles of the cell surface membrane result from properties of phospholipids? Okay, let's go. To allow cytokinesis. Okay, these are the statements. So which one is correct? So I want to pause the video. Okay, cytokinesis. So try this question first, guys. Cytokinesis is when the cells split into two. Okay, so it's due to phospholipid. So this is correct. Okay. So cytokinesis is possible because of phospholipid. So that's correct. All right. To allow entry and exit of oxygen. Yes, oxygen and carbon dioxide, they are non-polar, so they pass through phospholipid because phospholipid has non-polar hydrophobic core. So because of phospholipid, that's why they can enter. Right. That's correct. To allow the phagocytosis of a bacterium. So phagocytosis is when, right, a cell engulfs solid particle, right? So that is possible because of phospholipid. Phospholipid is actually flexible, so it will just fold inwards, okay, and then forms a vesicle. So it pinches inwards in order to form a vesicle and allow the molecule to move inside. So that means that 3 is also correct, okay? Yeah, so all of them are correct. They are the role of phospholipids. Is that okay? Yeah, let's go to the next question quickly. Yeah, says which factors can be changed to affect the rate of facilitated diffusion across a cell surface membrane? Which of the factors affect, okay, rate of facilitated diffusion? So pause the video and try it. So these are the factors that affect the rate of diffusion. Surface area, yeah. The higher the surface area, the higher the rate of diffusion. There's a positive, right, proportional relationship here. Surface area increases, rate of diffusion also increases. The concentration gradient, yes, the steeper, the higher. Number of specific protein channels. The more protein channels we need for facilitated diffusion, the more, all right, the uptake of the molecule. Is that okay? So 3 of them are correct. They all are affected. Okay. The diagram shows an experiment using a model cell to investigate the movement of substance. Okay. Yeah, that's the model, guys. Yeah. Which statements are correct? So in OBJ, you have to be careful, right, of the questions before you attempt it. Which statements are correct? Which statements are correct here, guys? Says, look at this. Inside here has 0.03 mole per dm cube. But make sure you pause this video and solve it first, okay, before you come back. So 0.03. So we have 0.03 sucrose here, okay, 0.02 glucose here, but outside we have 0.01 mole of sucrose. So we're going to have, right, more sucrose inside, and the sucrose cannot move outside because sucrose, can you see, this is 0.03, outside 0.01, so sucrose can move outside, okay. Sorry, it cannot move, yes, even though we have more here inside than outside, it can't because it's a disaccharide is bigger. But we see glucose will move because glucose is smaller. So when glucose moves, right, when it moves here, it reaches equilibrium, okay. So water can as well move, but glucose moves when it moves out, sorry, because it's moving from high concentration, because here is 0.02 to lower concentration, 0.01, okay, to reach equilibrium. So let's look at what is correct. There's net movement of sucrose. No, sucrose cannot move because it is big, right? It's a disaccharide. So statement 1 is wrong, okay. Number 2 says there is net movement of glucose out of the model. Yeah, it's going to move out of the model. This is the model cell. So this is the model cell. So it's going to move out of the model. Why? Because it's moving from high concentration to low concentration. So it's moving from high concentration, which is 0.02 inside here, right, to 0.01 outside. So yeah, so this is correct. All right. 3 says there's net movement of water into the model cell. So water is going to move because we have high concentration of sucrose there. Sucrose can move, so water potential inside here will be less. Okay, so water is going to move inside. Okay, all right, by osmosis. So 2 and 3 are correct. So 2 and 3 are correct. D is answer. All right, yeah, let's roll, guys. So the diagram shows partially plasmolized plant cell. So that's it, guys. What is found at Z? I want you to pause the video and try the question. So what is found at Z? Okay, what is found at Z here? This is a space. This cell is plasmolized. Solution X is inside. This was put into a solution Y. Okay, remember that this is cell membrane withdrawing from cell wall. So this is cell wall here. So when cell membrane withdraws from cell wall, okay, cell wall is fully permeable. So solution Y will enter. Cell wall is fully permeable. So solution Y will enter and fill up this place. So this Z will contain solution Y because cell wall is fully permeable. Is that okay? Yeah. So it can move inside. So the answer is C. Is that okay? All right. Single cell organism lives in fresh water. Water that enters the cytoplasm of the cells by osmosis. Okay. All right, you can see that single cell into a structure called, so moved, all right, into a structure, is collected into a structure called the contractile vacuole. So contractile vacuole helps to move water out of this single cell organism that lives in fresh water. Okay, fresh water has less solute, so water keeps moving in. Now to remove the water, the contractile vacuole, okay, the cell surface membrane, okay, you can see that. So that's counted the number of times that the contractile vacuole filled and emptied when the cell was placed in a solution with different water potential. The results are shown in the table. You can see that. So please, you always look at the table very well. Okay, so which statement explains the pattern observed as water potential of the external solution decreased? Okay, like you to pause this video and try it by yourself. Okay, that's how we roll. Pause the video, try it by yourself, guys. Now if you look at this, you can see that water potential of the external solution. Okay, remember, if we try to put a picture behind this, okay, let's assume we put it inside the beaker here, and this is the cell. It's an animal cell. Okay, fully submerged right into a solution. Okay, and we are varying the water potential. Okay, so if you see from the table, what does the table tell us? Can you see what's happening to the water potential? Water potential of the external solution. That's this external solution. This is the cell, this external solution. Okay, so it was 0 here. That means pure, no solute at all. Can you see? So if it was 0 here, that means no solute at all. That means that there's a steeper concentration gradient. No solute here, but more solute. So water continues to move in, right? Now, the rate at which water moves into the cell when it is 0 is high. That is why the rate of contractile vacuole emptying. So contractile vacuole has to, the rate is high. It has to continue to remove the excess water, because if it does not, the cell will swell up and burst. Okay, so can you see now? This is the highest, right, water potential of the external solution. That's 0, but you can see that it's becoming more negative. That means water potential, right, is decreasing. The more negative the water potential, the lower the water potential. Is that okay? So that means that the more negative becomes, the water potential here becomes right lower. So not too high compared to this, the water potential inside the cell. So because it's becoming lower, so the rate at which water enters the cell reduces. And that is why, can you see, the rate of contractile vacuole emptying decreases, because less water is now moving inside. So it continues to happen until you get to a point where water doesn't move inside. Okay, so we don't need to remove the excess water. Okay, look at option A. The question says, which statement explains the pattern observed as the water potential of the external solution decreased? Yeah, it's decreasing. A says the water potential gradient between the cell and the solution increased. Okay, it doesn't increase. So causing water to move into the cell more rapidly and to empty more frequently. Can you see that as it decreases, okay, water moves less rapidly? How do we know? The rate of emptying decreases. Okay, the rate of emptying decreases. The rate at which water moves in decreases. So the rate of emptying also decreases. So water does not move in more rapidly because the water potential is decreasing. Why? The external solution is getting more, right? It's getting more solute. That's having less water potential. A is wrong. Now B says the water potential gradient between, okay, the cell and the solution increased, right, causing water to move. Now it doesn't increase, so it's causing water to move into the cell less rapidly. Now this less rapidly is correct, but the water potential does not increase. Can you see that this is decreasing, right? So the water potential between this solution and the cell does not decrease. Okay, so but this says that it increased. So this is wrong. Okay, C says the water potential, okay, gradient between the cell and surface solution decreased. Yeah, this is correct. Yeah, because it decreased. Says causing water to move into the cell more rapidly. Now if it decrease, the rate of movement will be less, not more. So this is wrong too. Now this is the water potential gradient between the cell and the solution decreased. That's correct, because the water potential is decreasing, causing water to move into the cell less rapidly. Yes, less rapidly. Yeah, water moves in less rapidly. How do we know that water is moving less rapidly? Because the rate at which contractile vacuole emptying, okay, works is decreasing from 30 to 20, all right, until 0. So guess what? D is the answer we're looking for. Okay, all right, that's it, guys. Okay, so let's look at the next one. It says, okay, cells contain vesicles. Liver cells contain vesicles that have proteins in their membranes, which are specific for the transport of glucose. When these cells need to take up glucose, the vesicles fuse with the cell surface membrane. How does the uptake of glucose occur? Okay, I want to try this. How does uptake of glucose occur? Okay, yeah, how does it occur? Now, glucose, pause the video and try to solve it, guys. Try it first, okay? How does it occur? Glucose is a solute, okay? Is smaller molecule and is charged, so it cannot be exocytosis. Exocytosis is for bulky. Exo and endo, they are for bulky molecules, they're out. It's not diffusion, because diffusion, all right, or simple diffusion is for non-polar molecules. Glucose is polar, so it's facilitated diffusion. Okay, that's why it requires proteins, because it's polar. It will be repelled, so it requires transport protein. So that is called facilitated diffusion. The D is the answer there, guys. All right, a student set up an experiment to investigate diffusion. Okay, let's see what he set up. Okay, the agar, this is the dimensions: 1.0 cm, that's the length, width, everything 1.0 was stained uniformly with a water-soluble blue dye. Okay, the block of the agar was put into a test tube containing 10 cm cube of distilled water at 20 degrees Celsius. The intensity of the blue color of the water after 5 minutes was measured. Okay, that measured. Okay, it's a typo there. 4 other experiments, A, B, C, D, were then carried out using different numbers of agar blocks, different sizes of agar blocks, and different temperatures. All other variables were standardized. Okay, which experiment give a lighter blue color in the water after 5 minutes compared to the first experiment? Okay, which one gives a lighter blue? Right, so I want you to pause the video and try to solve this question. Okay, Pause the video. Now remember that they were put in a test tube, okay? Pause the video and see how you apply the concept you've learned, okay? They were put in a test tube, right? So don't want to draw them, okay? So okay, now they had light blue, okay? So that means that the blue is going to move out of them into the solution. All right, so remember that we were told that they had, okay, look at the type of blue they were mentioned, the uniformly with water soluble blue dye, okay? And of course we're measuring the intensity, okay, of the blue color. Now that means the question says which experiment would give a lighter, these are things you have to pay attention, lighter blue color in the water. That means the one that has lighter blue color here, okay, is the one that shows less diffusion, right? Less diffusion, right? Less diffusion. Okay, so which one shows less diffusion? Now if you look at option A has larger surface area to volume ratio. Can you see? Smaller length, this is 1 cm, this is 0.5, check. So, and also at higher temperature, so these ones can never have lighter blue color, because the diffusion will be more, it will be faster here compared to the original one, okay? And if you check this one and also check this other one, check C2, okay? Why? Because C has higher temperature, so higher temperature there will be higher rate of diffusion, and of course it also has smaller length compared to the original. But if we come to B and D, okay, now of course if you look at B, yeah, look at this, can you see D? Okay, let's go to B first. See B. So B has lower, of course, lower surface, it has higher surface area to volume ratio compared to the original agar blocks, okay? But it has, you see the temperature, this, how many of these were there? Okay, there are about 3 of them, okay? But this has about 2, did you get? And the temperature is 20, okay? So this is also 20 degrees Celsius, 20 degrees Celsius, okay? So if you look at lower temperature, okay, this was 220 degrees Celsius. Now if you check D, 2 of them are most likely the answer. Now if you check this, see that this has a higher surface area compared to this, and this is just 1, okay? So I think the one that will be lighter, because this has, even though it has lower surface area to volume ratio, but there are 3 of them, okay? Why there are 2 of these at the same temperature? So that means this is going to be higher because it has 3 of them, has 2 of them. This will be the least because it has longer length, okay, and just 1 number of agar, because number of agar blocks also affect rate of diffusion. So it's going to be 2. So after this, the one that have lighter should be this, okay, lighter blue color in the water after 5 minutes compared to the first. So it should be B, okay? All right, now look at the next one. Batrachotoxin is a poison found in frogs in the Colombian jungle. All right, the poison is used to produce poison dart, okay? The poison works by increasing the permeability of the cell surface membrane of nerve and muscle cells to sodium ions, which move out of the cell. So you see that the sodium ions move out of the cells of nerve and muscle, okay? It moves out. 4 students made a statement about how the poison affects the cells. So let's look at the statement. So the question is, which statements are correct for the cells affected by batrachotoxin? Now I want you to solve the question, pause the video and solve the question, okay? So look at it. Which statement? They say water leaves cells by osmosis, causing the cell shrink. Yes. See, now guys, when sodium ions leaves the cell, okay, when it leaves the cell, the water potential of the cell will increase, okay, and then causing water to also leave. So whenever solute leaves a cell, what happens is that water will also leave with it. So this is correct. So water leaves, because when solute leaves, the water potential increases. So water leaves by osmosis. Now number 2 says water enters cells by osmosis. There's no way water will enter, because sodium ions are leaving the cell. When they leave the cell, the water potential will increase higher than the water potential outside the cell. So water would leave, not enter. So this is wrong. 3 says when sodium ions move out of the cells, okay, the intracellular fluid has a more positive water potential, okay, than the extracellular fluid. Yes. So intracellular fluid has more positive means, okay, less negative, and less negative means higher water potential. So if this is a cell, so and this is the intracellular, the inside the cell. So when sodium ions leave, okay, so what happens here is that the water potential inside the cell here will have high water potential, which is more positive. Is that okay? Yeah, so this is correct. Now 3 says when the sodium ions move out of the cells, the extracellular fluid has a more positive. No, when sodium ions move, it will not have more positive water potential than intracellular. More positive means it will have higher water potential. Now sodium ion is moving out, so this outside will have low water potential. It's going to have more negative, not more positive. So 4 is wrong. Only 1 and 3 are correct, okay, which is A. All right, which processes use energy? Come on guys, let's rush this. Which processes use energy in form of ATP? Which of them? So yeah, choose the answer quickly. The ones that use ATP, endocytosis, pause the video and try it. So just endocytosis and exocytosis, they use ATP to form vesicles and the movement of vesicles, okay. Endo and exo, they use ATP for formation and movement of vesicles. Fast diffusion does not use ATP, so it's just 1 and 2. An indicator is colorless in acid and pink in alkali. It's pink in alkali. Okay, in an experiment, a petri dish of agar was prepared using an acidic solution of this indicator. Okay, a disc of agar 1 cm in diameter was removed from the center to create a well. A white card showing circular marker line 1 cm apart was placed underneath the petri dish. 1 cm cube, this is cube, 1 cm cube alkali solution was put into the well in the agar and the stopwatch was started. Okay, circular disc of pink color appeared and spread through the agar. It reached the first marker line in a short time, okay, but took longer. Took longer to reach the second marker line, and a very long time to reach the third marker line. Okay, so the first marker line was closer. What explains this observation? Okay, yeah, how does it move? Remember that they told us that the indicator colorless in acid, okay, pink in alkali. Pause the video and try it. Okay, pause the video. So it's the alkali that causes the pink. So any question that has to do with alkali here, we choose it. A mentions alkali, C mentions alkali, right? Alkali solution. All right, but you see, if it's going to move, it's going to be by diffusion, simple diffusion, not facilitated. So facilitated means it requires transport protein, but it's just moving through the disc agar. There's no cell membrane involved. Facilitated diffusion deals with cell membrane, but simple diffusion doesn't need cell membrane. It's just a movement. All right, so it's simple diffusion of the alkali because alkali forms pink solution, pink color. All right, the diagram shows a section of glycoprotein molecule found embedded in a cell surface membrane. Each of the amino acids is represented by a small shaded circle. Can see that? Yep. So let's go, guys. Yeah. So they say each of the amino acids represented by, so they want you to find out their property. So this is property. Pause the video and try it. Okay. Pause the video and try the question. Don't forget that you can see this amino acid Q. This is the amino acid chain forms an alpha helix, and they are forming the alpha helix inside the hydrophobic core. This hydrophobic core, where we have the tail of phospholipid. This is a phospholipid bilayer. Okay, this hydrophobic core. Hydrophobic core, right, is always, all right, is hydrophobic. So it's non. It allows non-polar. Whatever that will be, there will be non-polar. Okay, not polar because they repel water or water-soluble molecules. So A and B, non-polar. Now amino acid at Q, this is facing water. So this is facing water environment. It must be polar. So that's polar. Is that okay? So that's okay. Of course, it says which role property of the amino acid is found. So you can see that we have found the answer already. A. Long chain saturated fatty acid change from solid to liquid at high temperatures compared with short chain unsaturated fatty acids. Which fatty acid would be more likely to form triglycerides in mammals that live in cold climates? Okay, which fatty acid will be more likely? Okay, so this is repetition of that. So which of them will be more likely in cold climates? So I'd like you to pause the video and try to fry the fish, guys. Pause the video and try to fry this fish. Remember, guys, remember that in cold environment, okay, that the phospholipid molecule, the membrane should become more fluidy, okay, in cold environment. If they don't become more fluidy, okay, they will be frozen, and once they are frozen, the cell becomes impermeable and then it dies. Okay, so which of these increases permeability, makes it more fluidy? Okay, the longer the chain, right, the less of course permeability, or less fluid. So anything that has long chain can be the answer. Okay, because the longer the chain, all right, the more closely packed the phospholipid bilayer in the membrane. So if you go back to the membrane there, look at it. If you have longer chain here, the longer chain, they will be packed closer. So there'll be less gaps between them. There'll be less gap between them. Okay, there'll be less gaps, and if there are less gap, there'll be less fluid. But the shorter the chain, all right, the more fluid, and we need the ones that must be, okay, that'll be fluidy, so that the membrane does not get frozen. Okay, so shorter chain, but of course, the more unsaturated, the more fluidy. Okay, so in cold environment, we need short chain unsaturated. All right, the shorter the length, the more fluidy. The more unsaturated, the more fluidy, and we need more fluidy triglyceride or phospholipid, right, for cold climate. So that's why D is the answer. When animal cells are cultured, salt solution is added to keep the cells alive. What is the purpose of the salt solution? Why do we add it? So pause the video and try it. Why do we add salt solution? Right? We add the salt solution so that, pause the video and try it, to lower the water potential, so that the water doesn't leave the cells. Is that okay? Alright, so to maintain the sort of isotonic solution, water doesn't, and the cell does not burst. Okay, to make sure the cell doesn't shrink, it doesn't burst. A says to allow for the diffusion of salt, nope. To prevent diffusion of other ions, nope. To prevent net movement, yes. To prevent net movement of water into or out of the cell. Okay, so that cell remains the same size, it doesn't lose water, it doesn't burst. It does not to provide energy, the cell can power itself. So C is the answer, to maintain the shape, okay, of the cell. The cell doesn't die. The following are all processes that allow movement into cells. So these are all processes. Which process requires ATP? I think we saw this question before. Okay, phagocytosis, active transport, two of them require ATP. This requires ATP for formation of vesicles. This is for movement against concentration gradient. So this is not ATP. Okay, so that's one, and okay, two, they require ATP. Which features are required to allow for efficient diffusion? Likely to try it. So which allows efficient diffusion? Which of them increases rate of diffusion? Okay, which of them increases rate of diffusion? Large surface area, yes. Short diffusion path, yes. Maintenance of, Yes. So 3 of them, all right, affect rate of diffusion. The larger the surface area, okay, the shorter the diffusion path, okay. Of course, the faster the rate of diffusion. So if you maintain steeper the concentration gradient, okay, the faster. So 3 of them. Which features are correct for active transport and facilitated diffusion? Active transport and facilitated diffusion. Yeah, and which one fits them? Now pause the video and try. Which one? And pause the video and try. The movement of molecules and ions depends on ATP. Now this works for active transport, but it doesn't work for facilitated diffusion. Facilitated diffusion is a passive process. It doesn't need energy because charged molecules are moving from high concentration to low concentration. All right, so this is not for both of them. 2 says they are specific for one type of molecule or ions. Yeah, so the transport, if it is, especially when we're dealing with carrier protein, so the transport, so there's that specificity there. All right, and they're involved in transport of specific molecules. 3 says they use membrane proteins. Yes, they use carrier and channel proteins. So 2 and 3 are correct, of course. All right, look at this. The diagram represents 2 solutions P and Q were separated by partially permeable dialysis tubing. Can see that? Yeah. What will be the initial movement of the molecules 1, 2, and 3 between solution P and solution Q? So I'd like you to try the question, okay? Now, net movement from P to Q. So that means it's going to move from P to Q, right? And anyone that will move from P to Q is move from high concentration to low. So the one that is more in Q here is this molecule 1. So molecule 1 is there are more of it here compared to the P. So it's going to move from Q to P by diffusion. So molecule 1, option that has molecule 1, A and B chosen. Okay? From P to Q, that means the one that is more in P compared to Q. So which molecules are more in P? Okay, look at it. Molecule, can you see this molecule here? That's molecule 3. So there are more, there are about 1, 2, 3, 4, 5, 6. None is here. So it's going to move from P, high concentration, right here. So molecule 3. So B is likely the answer. So the one that is no net movement, the ones that are equal, okay, that should be this, all right, box here. That's molecule 2. Let's count it. 1, 2, 3, 4, 5, 6, 7, 8. 8 here. 1, 2, 3, 4, 5, 6, 7, 8. So they are equal. So there's no net movement. So the answer is B, of course. All right. A student weighed a cylinder potato and then put it into a test tube containing a salt solution. The potato cylinder was removed from the salt solution after 1 hour. It was blotted dry and then reweighed. The solution recorded that the potato had lost mass. Wow. So what type of solution is that? Which row shows the correct explanation for the result the student collected? Why did it lose mass, guy? Okay, it says A, but let's find out. Okay, water potential of the potato. Now if it's going to lose mass, what was the water potential of the potato cell before soaking compared to water after soaking? So before soaking, it has higher water potential, okay, so that after soaking it now has lower. It has lost water. That's why the mass decreased. So it has higher. So after soaking it became plasmolized because it's a plant cell. So that's why it's A. Okay, yeah, lost water. All right, which functions are performed by glycoproteins on the surface or cell surface membrane? So what are the functions of glycoproteins? Do they act as enzymes? So what is the answer? Okay, to act as enzymes catalyzing reactions, they do not work as enzymes, so 1 is out of it. Okay, to have specific sites where chemicals can bind. Yeah, that's cell recognition. So they act, okay, site for cell recognition. 2 is correct. To secrete chemicals used for cell signaling. So do not secrete anything. So it's only 2. Okay, so that's why C is the answer. So thank you guys for hanging out. Okay, so we want to know that we're having another series for you on mitotic cell cycle. So hang on and check our video, share, all right, and see you next class. Okay, this is Shedro. Bye.